evening. Yesterday, completed about the pollen grain structure and oval structure. So today we will discuss about the types of ovule. Types of ovule very important for short question. And structure of ovule, pollen grain structure, these are all important. And here the most important topic, let's see the types of ovule. Let me share the PPT slide here. Right. So first of all, let's see the types of ovule. This is very important. Types of ovule, very important for short question. So here, the given heteropus ovule, first heteropus ovule. Or heteropus or orthotropous ovule. Second one, anatropous ovule. Anatropous ovule. And third, hemianatropous ovule. Third one, hemianatropous ovule. Compylotropous ovule. Amphitropous ovule. Circitropous ovule. Actually, this is the evolution. So first, let's see the first heteropus or the orthotropous ovule. Any type of ovule, these ovules are classified based on the structure of the ovule. The structure of the ovule, especially by micropyle, based on micropyle. This is micropyle. 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 And body, this is called body, and base of the body called as chalaja. Base of the body called as chalaja. Chalaja. And the stalk, this base chalaja and the stalk. What extent? The stalk. This is called funicle. Stalk called as funicle. Already told you yesterday. Clearly explain about the structure of the ovule. And this is called as funicle. Funicle. So remember this micropyle, chalaja, and funicle. Based on this, we can classify the ovules. The first of all, first one, the orthotropous or anatropous, which is the most primitive type of ovule. In this, the arrangement of micropyle and chalaja and funicle are in straight line. They are micropyle, chalaja, base, funicle. They are in straight line. So this type of ovule called as orthotropous or anatropous, orthotropous or heteropous ovule. Example, betel, piper, and polygonum. They are the best examples. After that, and evolution, this is considered as the first one. And the second one, actually, this is hemitropous. Second, hemianatropous. And what happens second hemianatropous? The ovule body turns into 90 degree. Now it is completely 180 degree. Sorry. 360 degree, 90 like this. First, first, let's complete from the atropos. This is the actual first line, straight line. Now what happened? The body turns into 90 degree. This is 90 degree angle. Now what happened in emitropos? The ovule turns into 90 degree. Now this is 90 degree angle. Here 90 degree. 90 degree. Now funicle body ovule is right. Funicle here the funicle or body ovule is at right angle to the funicle. Example ranunculus. That means this funicle, this one. Funicle stand. This funicle stand, this funicle, this one. Right angle to the right angle, nothing but 90 degree angle to the body. For this body, 90 degree angle. And also 90 degree to the micropylar. Micropylar and chalaja not in same line. Micropylar and funicle not in same line. What happened? The same funicle, just imagine it is turns into 90 degree. Turns into 90 degree. So this type of called as hemianatropous, example ranunculus. The insert in our book given the example also. And next one, third, compylotropous ovule. Third one, compylotropous ovule. The simple in compylotropous, this 90 degree. Actually, this is 90 degree. In compylotropous ovule, what happened here? The ovule is some more right angle to the funicle. 90 degrees, some more 90 degrees. That means exceed the 90 degrees. Almost 120 degrees, 120 degrees. Almost 120 degrees. So in this situation, funicle. Now this is 120 degree. That means degree increased. 120. Now here the 90 degree. First straight line. Now this body, the micropylar with body turns into 90 degree. Now this 90 degree now reach somewhere down. Somewhat down. You can see the line here. It's 90 degree, but here somewhat down. As a result, it is the degree of the angle is 120 degree. Now this type of ovule called as compylotropous ovule. Best example Leguminosae and Crucifera. And here the micropylar end is bent down slightly. This micropylar here the straight, but here micropylar slightly down. So this type of ovule called as compylotropous ovule. And amphitropous ovule after third one. Now the fourth, this is actually these are the steps. Fourth one, amphitropous. You can imagine what happened in amphitropous. This amphitropous evolved or developed from the compylotropous ovule, which means the somewhere down, micropylar somewhere down like this. Micropylar reach the somewhere down. Micropylar reach somewhere down. And here the degree. A degree also, angle of degree also increased. Almost the micropylar and funicle somewhat close here. Now this degree 160 degree, 160. That means degree increased in curvature. 160 means curvature, more curvatures happening here. So this type of called amphitropous ovule. Example lemma and puppy. Sorry. Amphitropous, right. Lemma and alisma. Lemma and alisma. And next one, anatropous ovule. And fifth, this is advanced one. This is advanced one. We can imagine where are the micropylar and funicle. Micropylar, this is micropylar and this funicle, they are arranged side by side. and the angle is completely turns into 180 degree. 180 degree. Here the 90 degree and here 90 degree. Total here 190 degree. Anatropos, the angle is 180 degrees. So this is Anatropos ovule, the most advanced one. Most advanced one. This mainly found in the all type of angiosperms family. Now this is the development. First primitive one, Atropos, Orthotropos ovule, the straight line, they are straight line. That means funicle, micropylar, chalaja, they are in straight line. But later what happened? This body turns into 90 degree. And later this 90 degree increase third Comphalotropus. And now they more reached, more down the micropylar towards the funicle. We can observe in this diagram also. Now it is the body turns into 1 degree. And another important character in, I forgot actually, the Amphitropus ovule. This is embryo sac. The center, this is embryo sac. Embryo sac. You can find any type of ovule. The embryo sac is common, straight. Here also common straight. Here also straight. Here also straight. But if you see in Amphitropus, is it straight line? No. The embryo sac shows horseshoe shape. That means somewhat curve. Amphitropus embryo sac shows somewhat curve. This structure look like a horseshoe shape. And later, during evolution process, what happened? The micropyle, some more. That means the micropyle and funicle almost closely arranged side by side. Now the body turns into 180 degree. Now this is the advanced one, Anatropus. And last one here, Cercinotropus ovule. The simple Cercinotropus ovule. This micropylar turns into total round. That means 180 degree and again 180 degree. If this turns into 180 degree, where is micropylar? Now is 180 and again 180 means it turns into full. Now the micropylar goes up. Micropylar goes up. Now this total degree, how many? Now tell me the total degree here, the body degree. Now Anatropus 180. If 180 completely turns in 180 degree, the micropylar down. When it turns into full, now the degree of the Cercinotropus, how much? Is there Mariam?
Yes, sir.
Now tell me the total degree of Cercinotropus. How
80
much degree?
and 80 you said, no, sir?
Yes, 180. Actually, the previous 180 degree. The 180 degree, this micropylar turns full turn, take full turn. That means this down, here off, and here full. Total here off means from this, for example, a 180 degree ovule, only the micropylar turns this range, half range of the ovule. Now degree, if half range means it 90 degree, right?
Yes, sir.
And another 90 degree add, that mean it is turns into upright direction. Now total 180.
180 degrees, sir.
Yes, 180, total 360 degrees.
360 degrees.
Total 360 degree. So this is types of ovule with example. This is very, very important. Very, very important. And also in examination also, of course, in our book given different order, you can write either anyone. That means you can follow your book headings or you can follow this class headings also. But this class is by the ovule formation by evolutionary. From first Anatropus, degree increase 90, Hemianatropus, some more increase, Comphalotropus, some more increase, Amphitropus, and more increase Anatropus, and complete full total turns 360 degrees Cercinotropus. Cercinotropus mainly found in xerophytes, example Opuntia. So this is about the types of ovule. Is it clear? Types
Yes,
of
sir.
ovule clear, right? Now, the pollen storage, this is general. Pollen viability, you must have learnt in laboratory, practical training classes.
Yes, sir.
Already pollen viability completed in practical training classes, and you can write the same. And 8th unit, principle of hybrid seed production. This is also important for essay question, hybridization steps and modification of sex. These are very, very important. Let's see one by one. So first, what is hybridization? What is hybridization? What is how the hybrids are formed? Now tell me, what is hybridization?
Hybridization, when we cross
Yes,
between two genetically dissimilar parents.
right. So now the hybrid form. This process called as hybridization. Are you able to see the slide? Just wait.
Yes, sir. Yes, sir.
Hybridization steps. Right. So definition clear. The crossing between two genetically dissimilar parents. If the genetically similar, that is the common one. If the genetically dissimilar parents, what happens if you take the genetically dissimilar parents? What is the offspring? Offspring different trait are there, or the same trait produced in offspring?
Different traits.
Different traits are produced. So these different traits called as hybrid. New trait, that means new trait of brain. That new trait called as hybrid. This process called as hybridization. Definition clear?
Yes, sir.
Right. In nature, to create genetic variability and for its wider adaptation, different environment condition. No need this all. Actually, definition okay. I don't
Yes, sir.
want to waste what time on my time. And let's see the direct hybridization steps. In hybridization steps, first of all, as the definition, two parents need. That is first selection of two parents. Second, selfing of the parent. Selfing, crossing of the parents. These two steps are important. First, selection. How to selection the parents? First, the parents, we can select the desirable characters. Desirable characters, where it is?
Desirable genotypes is written, sir,
Yes.
in
Yes,
second line.
right. Desirable genotypes, nothing but genotype, the genes denotes the characters. Right? And this
Yes.
this would be more from segregated population. Segregated population means here what is meaning of segregation? If the segregation means, for example, segregation, for example, here this is tall character. Here dwarf character, tall one gamete, capital T, here the small t, another for example, this is the male, this is female. But after first fertilization, what happened? Capital T, small t form. Capital T, small t form. These two. But while the gamete formation, they are segregated, not entering into the same gamete. That means actually here the both characters together, both male and female together. Right? While the segregation process, capital T enter into different gametes and also small t enter into another gametes. So this is called as segregation. So these genotypes from the segregating population, and here the female parent must be selected from the local types. Female one must be selected from the local type because they are the best adaptable. Best female one easily adapted to the weathers. That's why we selected from the local type. While selecting the parents, all the important characters to be combined must be kept in mind. That means, first of all, you want to decide what character you need in coming generation. And that purpose, as such, you selected desirable character. Desirable character, how can we get desirable character? This desirable character getting by segregation of gametes. Right. So anyway, after selected the parents, now the selfing the parents. Selfing the parents. That means the selfing parent, that means the selfing means self-pollination. fertilization. Actually, naturally, self-pollination takes place. That is commonly occur in some plants. But selfing not to be done in self-pollinated crops. Here given clearly, self-pollinated crops actually as a characteristic nature they can self-pollination. But in cross-pollinated crops also we can promote to self-pollination by artificially. Let's see later artificial methods. So in second step, after selected the two parents already we selected the two parents to different genotypes. Already two different. Actually, two different genotypes, most probably there is no chance to self-pollination. They are mostly the cross-pollinated, but this cross-pollinated, for actually, do you want to get the desirable character more without changing? Without changing. For example, capital T, small t, one of the desirable character. One plant. Do you want to same? Would you like to self-pollination the same plant? When you self-pollinated same plants, the desirable characters carry forward for next. Each continuation for each and every generation. What happens if cross-pollination occur after getting desirable character? This is the desirable gene, desirable character. Do you want to this desirable character plants more? In that situation, you go through the self-pollination only. During self-pollination, the same genes carry forward for many years, many generations. What if cross-pollination occur? What if cross-pollination occur? Any changes
That
in desirable gene?
other plant traits will
Right.
come in the next generation.
Right, other plants characters will come. They may be
Yes.
desirable. They might be desirable, or they not might desirable character, undesirable. That's why there is don't give the scope for the cross pollination. We promote only self pollination. Even the plant type adapted to cross pollination, we forcefully, artificially, we promote the self pollination only. Because some plants only for cross pollination, they don't allow self pollination. There are some reasons. I don't want to go through that. But remember, some plants are self pollinated. They are naturally self pollination, no problem. But some of the plant, they are not self pollinated. They are the cross pollinated. The cross pollinated plants, we artificially promote the self pollination. Right? These two steps are clear.
Yes, sir.
Right. Now let's see how artificial self pollination takes place. So after selection, after pollination, only self pollination. In actually, if any cross pollinated type of plants are there, we forcefully entertain the self pollination. Let's see some self pollination methods here. Self pollinated methods here. Right. Are you able to see the slide? Artificial self pollination.
Yes, sir.
Right.
Yes, sir.
Artificial self pollination done by the first method, emasculation method. First method, emasculation method. What is emasculation? You must have heard this word. Emasculation. Emasculation means simple.
Yes, sir. We have done in the practical. Emasculation,
Right.
removal of male organs, anthers.
Removal of male organs in bisexual flower. Actually, what happened in bisexual flower? Bisexual flower means
Which is having two sex. Like
anther and gynoecium. If both are if the
androgen
both
and
organ
gynoecium.
right, if the both organ present inside the same flower, they, most probably promote the self pollination.
Yes.
And in some regions, some of the plants, cross-pollinated plants also have the bisexual flowers. Right. What in emasculation, what we done? very important step, hybridization. Before the removal of the male organs in bisexual flower before the anthers, before the anther ripe. Just wait. One minute. Okay, sorry for the inconvenience. Right. Emasculation. What is removal of male organs in bisexual before the anther ripe? Right. And shed the pollen on the stigma to prevent the self-pollination. So actually the heading is prevention of self-pollination, not artificial, prevention of self-pollination. So in bisexual flower, what happened? In bisexual flower, the male and female, both the sex organ present, there is a chance to self-pollination. But if you want to desire character, for example, A plant and here the B plant, A plant, actually this is the bisexual plant, but it has no resistance power. But B plant have the resistance power. But do you want to get this plant in next generation? In that situation, you forcefully prevent the self-pollination in A plant. That means remove the male organs. What happened? That there is no chance to self-pollination. Now you take the desirable character from the bee plant in the form of pollen grain Now this pollen grain falls on the stigma of the A. The what happened later in next generation, this B plant character carry forward to the next generation. So this process called as emasculation, and this emasculation done by different methods. The first one forced open method, second one clipping method, and ring cut method, and keel rupturing method. Let's see the first one, forced open method. In this method, the flowers are opened by force before it open naturally. Actually, before opening of the flower, we can open the flower forcefully with help of the forceps and remove the anther. This is best example in tomato, brinjal, and chilies. And clipping method. This technique is usually used to cereals like rice, wheat, bajra, this type plant. In this method, top 1/3 portion, 1/3 portion of the spikelet. Spikelet means inflorescence. Already told you. Spikelet means inflorescence are cut with scissor and anther are removed with the forceps. So the any method, these all methods under the emasculation process. The first emasculation, removal of male organs to prevent the silk pollination. That's it.
Sir,
So in this
can you tell once again clipping method?
clipping method simple. For example, do you know the cereals plants like rice and wheat?
Yes, sir.
In the rice and in this method, 1/3 portion of the spikelet. 1/3, for example, this is the plant, rice plant. And let's change color here. This is the inflorescence or spikelet. This is called inflorescence. Inflorescence or spikelet. So in this, the top 1/3 for example here, for example, this is the off 1/3. The cut 1/3 of the portion of the spikelet cut with the scissors, and the anthers removed. Anthers removed with help of forceps. Actually, the most of the in spikelets, the anthers, the male flowers above part, top part. If you cut the top region, what happens? Automatically, the anthers are removed. Only the female organs are present in the below place, under place. So this is clipping methods only follow in the cereals. Is it clear?
Yes, sir.
Next one, ring cut method. The ring cut name given itself, the circular cut is made by the base of the corolla at almost 2 mm away from the ovary. The corolla is removed and the anther is picked out with help of the forceps. Example, bendy and cotton and ligneous crop. So in this actually, do you know the corolla? Calyx corolla.
Yes, sir. Calyx.
Calyx, green parts, protection parts, corolla, attraction parts. So in this method, we made a circular cut, the base of the corolla. 2 mm away from the ovary. Ovary is also part of the flower. Now the corolla is removed and the anthers are picked out because actually this anther inside the corolla. Without separated the corolla, we cannot pick it up the anther. That's why the first we separated, we make a hole to the corolla base, and after that we picked up the anthers with help of the forceps. Example, bendy, cotton, and leguminous crops. And keel rupture. The keel in Papilionaceae family, Papilioideae or Papilionaceae flower, the most not only black gram, green gram, red gram, all the pulses crops, the keel petal is ruptured and stamens are removed because in this flower, in Papilionaceae flower, the anther, the stamens are protected inside the keel plant. If you rupture the keel petal, what happens? The stamens are exposed out. Now we can easily pick it up with help of the forceps. That is the keel rupturing method. Is it clear? These methods all preventing self-pollination preventing methods clear?
Yes, sir.
Right. In open, forced open, it is before mature of the flower, we can forcibly open the flower and picked up the anther or remove the anther. Clipping method make a clipping method 1 by thirds cut the spikelet. That means we can remove the anthers. That means male flower. Next ring cut method, the cut the corolla base 2 mm away from ovary. As a result, corolla we can remove the corolla. If we remove the corolla, we can easily remove the anthers also. And keel petal, already you know the Fabulosaceae family. In this different type of petals are there. Standard petal, keel petal, and harms petal. There are different type of petal. But inside keel petal, only inside the keel petal, all essential parts are protected. Essential part means anthers and stigma. Anthers and gynoecium, they are all protected in keel petal. If we rupture the keel petal, what happens? This reproductive organ exposed. When it is exposed, we can easily separate the stamens. So this is the methods are we promote prevent the self pollination. And another method is hot water treatment. Hot water treatment. Just wait. Hot water treatment. In hot water treatment, the name itself given hot water treatment, right? Just
Yes, sir.
1 minute.
Sir, hot water, cold water, chemical, alcohol, that is done, sir, already. We will see male sterility method and self-incompatibility
Where it is?
on page number 61, sir.
61. Male sterility method and self incompatibility. Right.
Yes, sir.
They also seem very simple because the name given itself male sterility. What is sterility?
To make incapable of germination.
The sterility, that means they are not able to fertilize.
Yes, they are not able to fertilize.
Right. So male sterility. So in crop like maize and jowar, bajra, potato, sunflower, male sterility occurs due to the cytoplasmic and genetic causes. Due to genetic causes, and also some more plants, the male sterility occurs by the environmental factor. That is another thing. But here in these plants, only cytoplasmic and genetic causes are both. But in this situation, emasculation is not needed. Is it need? Emasculation.
No, sir. No.
Already male sterility. Here this is no fertility. Sterility. No need to emasculation. Next, self-incompatibility. In crops like tobacco, cabbage, cauliflower, the pollination with the desired pollen is possible. Without doing emasculation due to the presence of self-incompatibility. Self-incompatibility means, for example, do how to tell? Just where? For example, let me take the simple example for self-incompatibility. Even though bisexual flower, even same plant pollen grains reach the stigma, but they can't fertilize. The same plant pollen grains not fertilized with the stigma. That means gynoecium. So this is called as self-incompatibility. Which are stamens and pistils in the same flower, and these structural organization of the bisexual flower increases risk of the self-pollination because of the genetic diversity, because of the gene, genetic character. So that is called self-incompatibility. In self-incompatibility also, no need to emasculation. That means actually what happened in this also, for example, we take here the tobacco or cabbage, cauliflower, we already given example in these plants, the same tobacco plant, the falls on the another tobacco plants, another tobacco plant stigma, or the cabbage, the falls on another cabbage, cauliflower same, another cauliflower, same plant, even same plants, but there is unable to fertilize with that plant because of self-incompatibility. So this process called as self-incompatibility. Is it clear? Hello?
Yes, sir. So they are failed to cross themselves like the flowers.
Right. In these flowers, in these plants also promote cross-pollination only.
Okay.
Self-pollination not occur and no need to, oh, that means what we call no need to
Yeah.
emasculation. Right. Emasculation. Right.
Yes, sir.
And the fourth one, given the bagging. Bagging
That is
also,
done, sir.
bagging,
Bagging,
crossing also
crossing,
clear. Right. Now,
crossing techniques. If we see once.
crossing techniques. Harvesting, okay. The emasculated flowers can be pollinated in different ways. Okay, first of all, you prevent the self-pollination by emasculated. Then how to cross-pollinate it? When the flowers are producing small quantity of pollen, it's transferred with help of the camel airbrush. Do you know the camel airbrush? You must
No,
have
sir.
used.
Camel hair.
Yes. Don't you actually during practical training classes, you have not done the section cutting?
Mm, this time
Section
we have
cutting.
not done the section cutting,
No.
sir.
Actually, this time means in 6th semester there is no section cutting. In 5th semester also no section cutting. Only section cutting in first year and second year. Only for first and second. That's it. Only
Yes,
first
sir.
and
I
second
saw.
year.
I saw the picture, sir, on the internet. That is
How?
a paintbrush, like
Right.
small
Right.
children
Right.
will do.
In during practical training classes, how can you select? How did you select the sections? After make a section from the given material, whatever it may be, the root or shoot, whatever
With
it
the help
is.
of the brush only, sir.
Right,
Camel
with help
brush.
of that is called camel airbrush. So in this technique also, after successfully completed emasculation, with desired pollen grain, um, carry with help of the camel airbrush and dropped on the gynoecium of the another plant. It is possible when the pollen grains are very small quantity, right? Now when the flowers are produced large amount of pollen, large amount, can you collect the large amount of pollen with help of simple tiny camel airbrush?
No, sir.
No, it will be collected the glazed butter paper bags. They are separated different type of bags and tested on the emasculated flower. Simple. Without instead of the camel airbrush, if the pollen grains are large amount, we simply collected with help of the glazed butter paper bag. If the glazed butter paper bag, the sticky nature, the pollen grains for sticky nature. When you dust it on the flower, it automatically falls on the emasculated flower, emasculated plant. That is second method. In case of clipping method, emasculation, this individual mature anther may be collected and inserted into the emasculated spikelet. The when did where did you see the clipping method? Clipping method present in. Hello?
Clipping method.
Which
Still.
plants? Cereal plants cut 1/3, right?
Yes,
One by
sir.
third portion. In this method, The individual mature anther may be collected in not at a time. In first and second time, whatever it is, little amount or huge amount, collected any at a time. But here the mature anther individually collected and inserted into emasculated plant, emasculated spikelet, because in these wheat plants, all the pollen grains, all the anthers are matured not at a time. And second, in fourth one, when the flowers are crossing numerous anthers and individual flowers, the anther number high in individual flowers collected after the corolla and calyx removed. So, if you collected the numerous anthers, the individual flowers is collected after the corolla and calyx are removed. What happens if you remove the corolla and calyx? Now the flower is opened. Now directly made it contact with the stigma of the emasculated plant. Example, bendy pomegranate. And last one, the inflorescence of male and female parents are enclosed in single bag and allowed for pollination. The seeds from female parents are harvested, raised in nursery. So in last method, male and female parents are enclosed in we take a single bag. In single bag, both type of parent inflorescence tied. And when they both are closely arranged, there is this chance to pollination. Now after pollination, seeds are formed. This is another technique for cross pollination. And last one, caging method. This is the special technique followed in for example in mango. In this method, the male and female trees are grown side by side. The female parent self-incapability at the time of flowering, both the trees are enclosed in an insect-proof cage net. Somewhat wrong here. Actually, the female parent is self-incompatible. At the time of flowering, both the trees are enclosed in insect-proof cage. Freshly reared houseflies released inside the cage. Right. In this method, what happened? Actually, in the mango, but cross pollination is entomophily done by the insect. To prevent the cross pollination, we enclose the flower with help of the cage, insect-proof cage net. What happens if you close the insect-proof? Is there any chance to reach the insect to the plant? If you covered with cage, no, there is no chance to approach the insect. Now freshly reared houseflies are released inside the cage. Later, we separately selected the different type of houseflies and released inside the cage. What happened here? Which facilitates the cross pollination. Right, the stones that are the collected. No need. So this is the caging method. So these are all techniques for cross pollination. Is it clear?
Yes, sir.
Now next one, modification of sex. So this is very, very important. Modification of sex, very, very important. Let's see.
Sir, tomorrow is the last class.
Yes. Tomorrow, last class, maybe how much time now?
Last 8 minutes are left, sir.
Okay. Let's complete the first this one. If any time I will talk with you. Right. So this is very important for long question. Remember it. So hermaphrodite, that means the bisexual flower, the both and male and female reproductive organs in single flower, right?
Yes, sir.
Hello. Right. In this
Yes, sir.
again, the complete flower incomplete flower, imperfect flower, perfect flower given. Complete meaning itself already you must have learnt in plant taxonomy third semester. Complete flower. Complete flower means flower containing all type of four rings such as sepals, first whorl, second ring petals, androecium and gynoecium. If you take the flower, the all sets are present, that is complete. The second one, incomplete. Incomplete means in this flower missing any one. Maybe calyx miss, maybe corolla miss, maybe gynoecium miss, maybe androecium miss. Imperfect. Imperfect means sexual distinctness. Monoecious versus dioecious. What is monoecious?
Single
Imperfect.
sex.
Right. Imperfect means they are separated with sexually. Monoecious and dioecious. Monoecious means both the male and female flowers on the same plant. Both the male and female flower on the same plant, that is called monoecious. Dioecious means male and female, male flower and female flower are different plants. That is dioecious. Perfect flower means the gamete maturation in different time. Production of the unisexual. So if the gamete formation, maturation in different time, that is called dichogamy. These are called, this type of flowers called as perfect flower. So anyway, again given here the monoecious and dioecious. Already told you the monoecious. The monoecious means the both the male and female flowers are the same plant. Right? Dioecious means male and female flowers are different plant. So male and female flower different plants means the plants also different, right? Male flower different, male plant different, female plant different. If
Yes, sir.
the plant have only male flower, that plant called as a male plant. If the female flowers plants called as female plant, that's it. But in monoecious, flowers are unisexual and are present in different position. Flowers unisexual, no doubt at all. Already told you, male flower and female flower. Male flower. Are you clear or can I explain with the diagram? Should I explain?
No, sir.
Right. Don't get confused with those words. So in monoecious, the unisexual flowers are different position on the same plant. Plant same, but different position. That is the monoecious. Right. Now, let's see the sex determination through the hormone chemicals. That means we can change the sex characters in the flowers with help of the hormones and chemicals. Let's see what are the chemicals and hormones here. Sex expression in dioecious and monoecious and dioecious plant is genetically determined. So whether it is dioecious or monoecious, it is determined by genes. And these also can be modified considerably extent by environmental. So this modification done by environment and some of the temperature. So there comes under the environment, photo period, temperature, phytohormones, next one, phytohormones, and there are some chemicals. What they given here. So here so remember the changing by the environmental condition.
Among them, phytohormone is very important, sir.
Yes, phytohormones, especially in phytohormones, the most important one are auxins and gibberellins. Auxins, and also gibberellin also. In auxin, especially naphthalic and acetic acid, naphthalene, acetic acid, NAA, ethanol, ethophan, cytokinin, But another one missing here, the axin with apart from the axin, gibberellin also, where they write here. GA3 means type 3 gibberellin. Hormones and chemicals inducing maleness. And some chemicals also given, silver nitrate. They are rare, abscisic acid rare. And other chemical also given here. So with help of these chemicals, we can modify the sex. That means male flower can modify it into female and female flower can modify it into male. Right? Is it clear?
Yes, sir.
Right, next one. And last one given the environment and sex modification. This is completely purely based on the environment. So environment has greater influence on the sex modification as far with phytohormones. Same. The phytohormones, actually the phytohormones can generate inside the plant naturally and can be applied also artificially. But in environment naturally occurs, but due to the introduction of photosensitive variations of hybrids and however in seed production it has its influence sex expression. Here given example cucumber, one of the example cucumber plant high temperature and long day length favors male flower. Actually cucumber plants maybe it is unisexual. Unisexual means the male and female different. If the same plant, that means the cucumber plant, if you apply the high temperature and long day length, 14 hours, you apply the temperature increase as well as the light intensity, light duration also increase. In that situation, the cucurbita plants only produce the male flower